Hello, welcome to General Embryology classes. So today's topic is on gametogenesis and spermatogenesis, where we will see the production of what is gametogenesis and production of sperms. So the gametogenesis, it is the formation of the gamete from the primordial germ cells and involves in the cell division, mitosis and meiosis. It involves cell division, mitosis and meiosis, which is an important cell division happens only in gametes. So gametogenesis means gamete formation. So they are these gametes are called as primordial germ cells. So primordial germ cells are derived from the epiblast cells, which are involved in the cell divisions of mitosis and meiosis. And these PGCs or primordial germ cells, which are derived from the epiblast, they migrate to the endodermal wall of yolk sac, and during 4th week of intrauterine life, and reach in an indeterminate gonad by 5th week of intrauterine life. So they migrate to the endodermal wall of yolk sac 4th week, and the aberrant migration leads to teratoma, of course. So aberrant migration may lead to teratoma or germ cell tumors, and the teratoma may arise from the primordial germ cells or from the epiblast cells, which are pluripotent cells, and therefore within the teratoma it present derivatives of all three germ layers, because epiblast will give rise to all three, that is ectoderm, endoderm and mesoderm. So ectoderm gives rise to epidermis of skin. So you may find in the teratoma skin, bone, teeth, gut tissue and so on. So that was about the teratoma. I hope you now understood from where the gametes are formed. Gametes are formed from PGCs, primordial germ cells. From where the primordial germ cells are derived? They are derived from the epiblast cells. So if you see the initial beginning stage of the embryo, first it forms a cluster of cells which are called as morula, and this morula, the outer cells will form the trophoblast. So here we can see the peripheral cells are called cytotrophoblast or trophoblast cells, which later on develops into placenta. And here we can see the inner cell mass, it develops into two layers. It arranges itself into two layers. The blue colored cells are the epiblast cells. These are called as epiblast cells. And yellow colored cells, what you are seeing, which are cuboidal, are the hypoblast cells. So the epiblast cells are very important. These are columnar cells and they are highly pluripotent stem cells. And these epiblast cells will give rise to primordial germ cells. And these primordial germ cells, once they are formed, they travel along the yolk sac. So here we can see the sac which is present below the endoderm. This is the yolk sac, and we can see a small amniotic sac which start developing here within the epiblast cells. So along the dorsal wall of the yolk sac, these primordial germ cells, they travel and they reach the indefinite gonad, and they reach the indefinite gonad when after the 3rd week only, because after the 3rd week we will have all three germ layers, ectoderm, mesoderm and endoderm. So mesoderm, that is intermediate mesoderm, will give rise to genital system. So at that point where we see the primitive gonads which are indifferentiate, we don't know whether it is a male or female gonad, that is called as indifferentiate gonad. So these cells will go and settle in the indifferentiate gonad and they will be in dormant stage. Okay. So that is, suppose aberrant migration of these cells will lead to some tumors called as teratomas, and teratomas usually contain all the three germ layers derivatives like skin, bone, teeth and gut tissue and so on. So that was about the gametogenesis. Now we know what are PGCs, primordial germ cells, which are the cells which give rise to male and female gonads. Primordial germ cells, we cannot differentiate it is male and female at that stage. So today's topic is on spermatogenesis, where we are going to see how the sperm, male gamete, how it develops. So spermatogenesis is the process in which the spermatozoa are produced from the spermatogonial stem cells. Spermatogonial stem cells are derived from primary germ cells, PGCs, and by the way of mitosis and meiosis. So in previous session you might have seen what is mitosis, meiosis and cell cycle. So now I can easily explain. So here we can see the stages of spermatogenesis. So the primordial germ cells, they remain dormant in the seminiferous tubules of the testis until puberty. They doesn't undergo any division. They are just in the form of PGCs only till they reach the puberty. At the puberty, they undergo a series of division to form spermatogonia, so which is called as spermatogenesis. So the primordial germ cells from the spermatogonia which yield the primary spermatocytes is by mitosis. So you see here, till the primary spermatocyte, we see only mitosis only. So only by means of mitosis, the PGCs, primordial germ cells, will turn to form primary spermatocyte. So first they turn to form spermatogonia, type A, type B spermatogonia, and from them we see the primary spermatocyte. So the primary spermatocyte divides meiotically. Now meiosis starts. Meiosis, we know it is in two phases, meiosis 1 and meiosis 2. So meiosis 1 is the critical stage because the reduction of chromosomes happens. The diploid number of chromosomes becomes haploid in meiosis 1. So the primary divides to form two secondary spermatocyte, and these secondary spermatocyte by meiosis 2, now they enter into meiosis 2 to form spermatids, which are round cells with having heavy amount of cytoplasm like a normal cell it is, that is called spermatid. Thus the primary spermatocyte gives rise to two secondary spermatocytes, and two secondary spermatocytes is subdivides to form four spermatids. So from one primary spermatocyte we get four spermatids. And meiosis has two stages. We know meiosis 1 and meiosis 2. So meiosis 1 is the actual meiotic division, where the meiosis 2 is similar to mitosis. It is an equational division. The spermatid, now it contains the haploid male gametes that results from the division of secondary spermatocytes. So the secondary spermatocytes divides to form spermatids. So as a result of meiosis 2, and each spermatid contains half of the genetic material present in the original primary spermatocyte. And round spermatids undergo further maturational events where it will shed off all its cytoplasm, and it gains its peculiar shape, this having head, neck and middle piece, and with the tail, that is called as spermatozoa or sperm. So finally the spermatid will turn to form spermatozoa. So this process is called as Permiogenesis. Where the spermatid turns to form spermatozoa, it is called as Permiogenesis. And the initial stages of spermatogenesis takes place within the testis or the seminiferous tubule. So we can see the testis here, the cross section of testis, where we can see a cut part of seminiferous tubule, and within the cut part of seminiferous tubule, the basal cells, the peripheral cells are spermatogonial cells and primordial germ cells, which eventually, these spermatogonial cells eventually divide to form primary spermatocyte. Primary spermatocyte enters into meiosis 1 and divides to form secondary, and as it moves to the lumen, towards the lumen, they form the spermatids and the sperms. We can see the tails of the spermatozoa. So till here. So the initial stages of spermatogenesis, it takes place here, and now these spermatozoa, they get detached from the large columnar cells which are present in between these cells called as Sertoli cells. They get detached and they enter into the epididymis. We can see from here the retitestis is there, and we can see the efferent ductules. Some 10 to 12 efferent ductules open into the head of epididymis. So where in the epididymis we can see the developing of gametes. So the gametes mature and gain the progressive motility, and they are stored until ejaculation. So spermatogenesis takes 74 days to complete. Total process of spermatogenesis, it takes 74 days to complete, and if the transport through the ductal system is included, it takes 3 months. So that is the total explanation about spermatogenesis. Here in this image we can see the sequential way how the spermatogonia forming in to form primary spermatocyte, primary divides to form secondary spermatocyte, secondary spermatocyte further forming into spermatids, spermatids are turning to form spermatozoa by spermiogenesis. So this is the basic idea. So the transit of the sperm through the epididymis is thought to be up to 12 days, and though the sperm can be stored in the epididymis for several weeks or months, there are 3 subtypes of spermatogonia in humans actually, which develop by the mitosis. Type A, that is dark cells with dark nuclei, that is the type A spermatogonia, and these cells reserve the spermatogonial stem cells which do not usually undergo active mitosis. So dark type A which doesn't undergo, and type A pale, dark pale type A or light color, light type A or pale cells with pale nuclei. You can see here with pale cells with pale nuclei, and these are the spermatogonial stem cells that undergo active mitosis. So these cells divide to produce type B. So type A spermatogonia will give rise to type B spermatogonia. Type B spermatogonia which divides and gives rise to primary spermatocyte. Remember one thing, till the level of primary spermatocyte it is considered as mitosis. Only mitosis happens. Only the change between the primary to secondary spermatocyte, that is the stage of independent assortment where we see the meiosis, where the mother and father characteristics can transfer equally to any child. So independent assortment takes place only in meiosis 1 division. So these things to be remembered. So each spermatid gradually changes to become a spermatozoa or a sperm. So this transformation of a circular spermatid, initially the circular spermatid into the sperm, is called as spermiogenesis. Thus from one primary spermatocyte there are 4 spermatozoa are formed, and 2 with 22 autosomes and 1 X chromosome, and that is 22 plus X, and the 23X, then the other 2 with the 22 autosomes and 1 Y chromosome. So 22 plus Y, that is 23Y. So we can see the matured sperm parts head with the DNA in the covered by a cap called as acrosomal cap, which is rich in enzymes, and the middle piece which contains mitochondrial sheath. Then the tail, the core, contains the microtubules. So this is the structure of a normal sperm. So spermatocytogenesis, it is the process where the primary germ cells undergo series of mitotic division to form a large number of spermatogonia, and depending upon their appearance there are 3 types of spermatogonia. Remember, dark type A spermatogonia and light type A spermatogonia. We said light one is active in mitosis, dark one is inactive in mitosis. This type A will again divided into dark and light type B spermatogonia. In this also the light type B are more active in mitosis, which further derive to form primary spermatocyte. So finally we get 4 cells, 2 with 22 autosomes and 1 X chromosome, and 2 with 22 and 1 Y chromosome. So this is the chromosomal complement. So here is the histological image of a seminiferous tubule, where we can appreciate the spermatogonial cells, that eventual stages forming a spermatocyte or spermatozoa, and we can see the Leydig cells here in between the seminiferous tubules. Leydig cells are outside, they are interstitial, they are present in between the seminiferous tubules, which produce testosterone hormone. This is the process of spermiogenesis, how a round cell turns to form a matured sperm. So morphological transformation of spermatid to mature sperm is called as spermiogenesis. Structure of a spermatozoa, which I already said, usually it is around 50 microns in length, consists of head and neck, constricted part, then middle piece, neck, this is the tail. So these are the parts of the sperm. The tail is further divided into 3 parts, the middle piece, then the principal piece and the end piece. So this whole thing is considered as tail, and head, neck. Spermatogenesis duration, usually I said it is 64 to 74 days. So spermatocytosis 16 days, meiosis 1 8 days, meiosis 2 16 days again, spermiogenesis 24 days. So total it comes to 64 to 74 days. So spermatogonia type A and B contains diploid number of chromosomes and spermatocytogenesis, mitosis. We see the mitosis. Primary spermatocyte, still there contains diploid number of chromosomes and it enters meiosis 1, and it results in the formation of 2 secondary spermatocytes which contain haploid number of chromosomes. So that thing you have to note, from primary to secondary results in spermatogenesis meiosis 2. Then 4 spermatocytes form spermiogenesis. This is meiosis 2 again. Then functional sperm, that is spermiogenesis, which finally the sperms are released, is called as spermiation. The motility of the gaining of the motility of the sperm is called as spermiation, which happens in the epididymis. Let's talk about some applied aspects of spermatogenesis. So non-disjunction, we know during cell division, the chromosomes lie in the equatorial plate. So when they are separating during metaphase, what happens? One chromosome may fail to separate, disjunction, non-disjunction, and resulting one cell group containing an extra number of chromosome, which is called as trisomy, and one cell group containing less number of one chromosome less, that is 45 chromosomes, it is called as monosomy. So trisomy in males, because of an extra chromosome, result in Klinefelter syndrome and abnormal gametogenesis also. And male infertility is one condition associated with spermatogenesis. Oligospermia, less number of sperms, that is 20 million per ml. Azoospermia, nil sperm count, and dispermia, triploid embryos, and pre-selection of embryos, and cryopreservation of sperms. So these are the. So here is due to non-disjunction, we see an abnormal chromosome, an extra chromosome at 21, which is 47 chromosomes. XX means female, plus 21 means it is an extra autosomal chromosome, Trisomy 21 is called as Down syndrome. So this is a karyotype of Down syndrome. Then factors affecting for spermatogenesis: smoking, alcohol, pollution, endocrine disorders, drugs, stress, genetic causes are the predisposing factors which may affect the spermatogenesis in males. And abnormal sperms, abnormality of sperms is a common as compared to the oocytes. So morphologically for clinicians, the sperm consists of two parts, that is head and tail for clinicians. And types of abnormalities are like head and tail of the sperm may be abnormal, having two heads, we can see here, having two heads or having two tails. And sperm may be giant, abnormally giant, very large sperm. This is a normal one. So giant sperm, or abnormally dwarf, very small sized sperm. And sperms may be joint or immotility and causing like potential fertility. 50% of the sperms should be motile after 2 hours of ejaculation, and some should be motile after 24 hours, and that is the normal timing. And genetical abnormalities like sperm having abnormal chromosomal content, which is rare as compared to oocyte. So abnormal chromosomal content, having one extra Y chromosome or extra autosome like 21 or extra chromosome like 13, Patau syndrome, Edwards syndrome, or trisomy 18 is Edwards syndrome. So all these are abnormal chromosomal content, or it can be sometimes some deletion and mutation having an abnormal chromosome within it. So let's see some questions now. Meiosis occurs in, I hope you can answer. Epididymis, seminiferous tubules, vas deferens, seminal vesicles. So of course the answer is seminiferous tubules. So initial stages of spermatogenesis, meiosis takes place within the seminiferous tubules of testes and progress to the epididymis, where the developing gametes mature and gain the motility and are stored until the ejaculation in epididymis. So here is the seminiferous tubule. So this completes the spermatogenesis, and we shall see with the next session on oogenesis and menstrual cycle. Bye.